was that the science is advancing incredibly quickly. And we're seeing therapies that have the potential to truly change outcomes for patients, including some who previously had very few treatment options. At the same time, there are still important questions about how we develop these therapies efficiently, how we manufacture them, how we design clinical trials, and ultimately how we make sure patients can access them. Many of those discussions at that meeting were fairly technical in nature, but obviously have real implications. Decisions about trial design, evidence generation, manufacturing, and regulatory expectations all affect how quickly promising therapies reach patients and who is able to benefit from them. And so that's why we wanted to bring this conversation to you all today. Today's discussion is an opportunity to unpack some of those topics, talk about what they mean for a patient, advocates, caregivers, and hear directly from experts and advocates who are helping shape the future of this field. We're fortunate to have a terrific panel joining us today. But before we get there, I'd like to invite my colleague, Elena Levi-D'Ancona, who will help set the stage with a brief overview of the landscape and some of the themes that emerged from the May meeting. So Elena, I'll turn it over to you.
Thanks, Mark. So thanks everyone for joining today. My name's Elena Levi-D'Ancona, and I'm a science policy analyst at Friends of Cancer Research. To start our webinar, I'll provide a brief overview of cell and gene therapies, highlight some of the unique characteristics that make their development particularly challenging, and summarize key takeaways from our May 6th meeting that will help frame today's discussion on opportunities to streamline cell and gene therapy development while maintaining patient safety. When we talk about cell and gene therapies, or CGTs, we're referring to treatments that use or modify a patient's cells or introduce genetic material to treat, and in some cases potentially cure, a disease or cancer. Broadly, cell therapies fall into three categories. Cell therapies, which use living cells from either a patient or a donor. Gene therapies, which alter or introduce genetic material within a patient's cells. And cell-based gene therapies, which combine both approaches. One of the best known examples is chimeric antigen receptor T cell therapy, or CAR T cell therapy. This figure illustrates the CAR T treatment paradigm. T cells are collected from a patient, genetically engineered to recognize a target on cancer cells, expanded to create sufficient quantities, and then delivered back into the patient. These therapies have been great and produced remarkable outcomes for many patients, in large part because they can be tailored to an individual patient's disease. However, that same personalization is one of the features that distinguishes cell and gene therapies from traditional therapies and can make their development more challenging. Unlike conventional small molecule drugs, cell and gene therapies are highly individualized, which can make it difficult to establish standardized development approaches and compare experiences across programs. Additionally, clinical development is often associated with greater protocol complexity, including specialized eligibility criteria, dose escalation strategies, safety monitoring requirements, and trial designs that must be tailored to the product and patient population. And manufacturing also introduces unique challenges. Cell and gene therapy manufacturing often involves multiple specialized facilities, complex logistics, and timelines that differ substantially from those of traditional drug development. And these considerations can influence both development strategies and trial design for cell and gene therapies. Because manufacturing was a major focus of both our working group discussions and May 6th meeting, I'd like to spend a few minutes discussing chemistry, manufacturing, and controls, or CMC, and what that entails. CMC refers to the information sponsors submit to demonstrate that a product can be manufactured consistently, safely, and reproducibly. For traditional therapies, this generally includes analytical methods, manufacturing processes, product specifications, and batch manufacturing data. For cell and gene therapies, however, CMC considerations are more complex. Many cell and gene therapies use autologous starting material, meaning each product comes from a patient's own cells. This creates variability in the starting materials, results in smaller batch sizes, and requires manufacturing processes that are individualized, time-sensitive, and often distributed across multiple locations. Using CAR T therapy again as an example, cell collection typically occurs at a hospital or treatment center. Those cells are then transported to a manufacturing facility for genetic modification and expansion before being shipped back to the clinical site for administration to the patient. These manufacturing realities create challenges that are not fully addressed by development frameworks originally designed for traditional therapies. Importantly, the FDA has demonstrated a willingness to apply regulatory flexibility to support cell and gene therapy development. Over the past several years, the FDA has issued a number of guidance documents aimed at adapting existing frameworks to the unique needs of these products. Examples include the 2023 guidance on manufacturing changes and comparability, the 2025 guidance on innovative clinical trial designs for small populations, the 2026 guidance on the plausible mechanism framework for individualized therapies, and the 2026 guidance on CMC flexibilities for cell and gene therapies. While these efforts have enabled meaningful progress, challenges still remain. The issue is not necessarily a lack of flexibility from the FDA. Rather, sponsors often face uncertainty regarding how to interpret and operationalize available flexibilities, and also uncertainty regarding whether those approaches apply to their specific products. This uncertainty can have significant downstream consequences, including delays in patient access, increased development costs for sponsors, repeated review cycles, duplicative data generation, and a growing trend of early phase studies being conducted outside the US, limiting US innovation and competitiveness. To better understand these challenges, we convened a working group that developed an issue brief identifying areas where targeted proposals and additional clarity could help streamline cell and gene therapy development. 3 key areas emerged, which were discussed at the May 6th meeting. First, in early phase development, establishing a common framework could reduce uncertainty by identifying which protocol elements can be standardized and which would warrant product-specific approaches, allowing sponsors to focus resources on the unique aspects of their products. Second, for CMC, a staged specification framework could provide a more structured approach to managing manufacturing changes across the full product lifecycle. And third, in clinical trial designs, additional clarification regarding when alternative trial designs and comparator approaches may be appropriate could facilitate more efficient development. Ultimately, patient-centered, risk-based regulatory flexibilities have the potential to accelerate access to innovative therapies while maintaining the high standards of safety, quality, and effectiveness that patients depend on. And with that, I'd like to thank you all for your time and pass it on to Rosanna Ricafort to introduce our panelists.
Thank you, Elena. So it's my pleasure to be with you all here today with this outstanding panel. We have with us Dr. Nirali Shah, who's a physician scientist in pediatric hematology oncology. She is internationally recognized as a leader in cell therapy and pediatric leukemia research. She's been a pioneer of innovative therapies in this field, including cellular therapies or CAR T cell therapies for relapse refractory pediatric leukemia. She's from the National Cancer Institute, and we're so pleased to have her on the panel to represent the voice of academia. We also have with us Dr. Lola Fashion-Aye. She is also a physician scientist, medical oncologist, regulatory leader. She has served in senior roles at the FDA, most recently in the Center for Biologics Evaluation and Research, or CBER, which does review all cellular tissue and gene therapy products. So we're pleased to have her. She's currently at AstraZeneca leading their regulatory efforts, and it's wonderful to have her on this panel to represent both the industry perspective, but also give us some perspectives from her time at the FDA. And last, but certainly not least, we have Tony Newbern, who is our Friends advisory advocate. Tony is nationally recognized as a leader in patient advocacy. He is a long-term survivor of high-risk multiple myeloma, and he reminds us all why we are here today. And so grateful to have Tony on this panel representing the patient voice. Thank you to Friends of Cancer Research for bringing this panel together around this important issue of really how can we help promising cell and gene therapies reach patients more efficiently while maintaining the rigor, the safety, and the evidence needed to support responsible development. It's not like, as Elena said, it's not about sacrificing rigor, but it's about really how can we apply that rigor in a more efficient manner so that we can develop our medicines to reach patients faster. This builds on that May 6th public meeting. You heard Elena recap key takeaways from the three main themes there for early clinical development, CMC, and clinical trial designs. A common theme emerged, and what we know in the cell and gene therapy space is that these therapies hold tremendous promise for patients. We've seen the potential to transform the treatment paradigm in hematologic malignancies, and in some cases deliver cure. But there are opportunities to continue to innovate, and the science is moving really, really fast. And it would be good also for the regulatory pathways and policy to move as fast as the science is moving as well. So the challenge is not often whether there's regulatory flexibility. We know that there's regulatory flexibility, and as a sponsor, we've seen that regulatory flexibility exercised. So it exists in principle, but how that flexibility is applied is something that more clarity and consistency across programs would really enable efficient drug development and timely patient access. So with that, let's get into it, starting, as we always do, from the patient perspective. So Tony, I'll pose the first question to you. What does it look like from a patient perspective to seek out and enroll in a cell and gene therapy trial? And where do you think the biggest practical barriers show up?
Thank you so much, Rosanna. Many people assume that once a promising trial exists, patients simply enroll. Unfortunately, it's rarely that simple. First, patients have to know the trial exists. Then they have to determine if they're eligible. Once after that comes the practical challenges, travel, lodging, insurance questions, time away from work, caregiver support, and financial concerns. And having gone through CAR T therapy myself, I can tell you that one of the biggest challenges is uncertainty. Patients often wonder, will my disease stay under control long enough for me to receive treatment? For many patients, especially those living in rural areas or receiving care outside major academic centers, access can feel just as challenging as the disease itself.
That's really helpful. Thank you, Tony, for sharing that. And indeed, patient access and that uncertainty of whether you will be eligible for a trial and whether the logistics and all the concerns that you raised will enable you to receive that is one thing. There's also a lot of times there are different phases of clinical trials depending on the program and the that's moving forward. And if we start from the earliest phases of a clinical trial, there's often anxiety about how those early phase clinical trials make it to patients. And so maybe Lola, if I can ask you to weigh in here and just share perspectives from a regulatory perspective about how the FDA greenlights a study to move from all of the preclinical evidence and pre-IND readiness that we do to make it to those first-in-human clinical trials, and maybe shed light on how those interactions between the FDA and the sponsors affect when patients can enroll. Because I think Tony raises an excellent point about that uncertainty, timely access, and we know that opening up clinical trials in a timely manner so that they can reach patients is as important as well.
Sure, I'm happy to provide some perspective on this, and just to briefly thank Friends of Cancer Research for convening this meeting. It's really great to have an opportunity to talk to the audience that we all serve here. I think it's important to just take a step back and just describe for the audience the mechanism by which a new drug or a new cell therapy that has not been approved by FDA can be studied in patients. The mechanism to do that is under what it's called an investigational new drug application, or an IND. This is the way in which a sponsor, whether that's a pharmaceutical company, a biotech company, an academic center, or some other party that's going to conduct a trial, seeks regulatory approval to allow their study to be conducted. And so there are certain data that are important to provide in that investigational new drug application, or IND application, to satisfy regulatory requirements. In early clinical studies, the primary focus of the FDA review is on safety, is on ensuring that this new drug or this new cell therapy that has not previously been studied in humans has the appropriate level of data information acquired through studies that have been done either in animal studies or other kinds of models that help us to predict what is a safe or reasonably safe dose to start investigating the new drug or new cell therapy in humans. And that there is sufficient information around the actual investigational therapy to enable safe administration to patients. And so that's where the FDA's review is focused on. Now, a company or a academic center could engage the FDA prior to submitting this investigational new drug application in what is called a pre-IND meeting to get agreement and feedback from the FDA regarding the quantity and quality of data that is collected in the preclinical setting, and information that could be leveraged from other similar trials and other similar programs to use to support the safe initiation of their study in this new product. And that can happen in a pre-IND meeting. It can even happen even earlier than that in what is called an interact meeting, where you're really sort of talking about the concept, the trial concept. So there are multiple opportunities to engaging with the FDA to determine what data is needed to start this trial, and to facilitate a speedy review when an IND is ultimately submitted to the FDA. So when that IND is submitted, it takes 30 days for the FDA to make a determination regarding whether it's safe to proceed in patients, or whether there are additional information or additional measures that a sponsor needs to take to provide that reasonable assurance of safety. So that's briefly what happens. So once FDA reviews it within a 30-day period, it's a go or no go. And if it's a go, then the sponsor then goes back and needs to engage local institutions, local sites, on what is needed to actually start those trials. And those questions are happening often in parallel, so it's not sequential. But there could be delays at any point along the way there that can really make it challenging for a patient to enter a trial. So what I just described is the regulatory piece, but there's also some processes and that are needed at the site level to allow a patient to be on a study.
Thanks, Lola. And maybe Nirali, I can call on you next, because that was a perfect kind of entree into from the sponsor perspective. And Nirali, as an academic sponsor, you heard Lola talk about safety, obviously, as everyone's primary focus, especially when we're talking about first in human trials. How can the development process that Lola described around the pre-IND and interact meetings and the negotiation back and forth with the FDA to provide the right amount of information to protect participants so that we can start early clinical trials. Can maybe you talk about where there's opportunity to conduct that process more efficiently?
Yeah, absolutely. And I'm really grateful that the FDA has these opportunities for having these early interactions. I think especially when you're doing a first in human cell or gene therapy study, or even a first in child, having that early input really allows you to incorporate the feedback in a meaningful way that would help, actually, make that FDA, getting the okay to proceed a little bit more efficient because you've already incorporated some of the questions and provided answers that may come up. I think on the clinical trial front, as Lola mentioned, getting the okay to proceed from the FDA is just one part of it. The next part is, you're going to the institutional review boards to getting the approvals to also do a conduct of analysis of the safety and risks that are to patients. I think in that regard, there are opportunities to think about having very set templated languages that it relates to, for instance, CAR T cell therapy, right? So that every time, what you want to avoid doing is having to start from scratch every single time. And I really like to think about the approach of iterative development. What can we take from, that prior knowledge that we've gained, incorporate into our new consents, having it be meaningful, and add to that what we don't know about this particular new study, because there's a long list of things for sure, but what do we know from our experiences that we've already had? So I think that definitely is an opportunity. I think as you think about multi-center trials, there's probably an opportunity there to also think about ways to harmonize or to streamline processes, whether it's using, centralized IRBs or having contracts in place that would allow you to have reliance agreements that are a little bit more feasible and make the process of implementing trial a little bit more time efficient.
That's great. And I want to pick up on a couple of things that you talked about. I love the idea of iterative development because oftentimes we are not starting from scratch and we don't need to reinvent the wheel. And you touched upon something. And I want to elaborate on the idea of a common framework, meaning, can we take the prior knowledge that we've developed and standardize some parts of early phase trial designs across programs and what benefits that could bring for patients and where might tailored approaches still be needed? So maybe if you can elaborate a little bit on that, Nirali.
Yeah, and I certainly also mentioned this during the prior meeting. I love the idea of having some harmonized foundation for dose-limiting toxicities, for eligibility. Again, these may have to be disease specific, but if you're doing a new cell and gene therapy trial, for instance, in acute myeloid leukemia, where there is a manuscript that was led by some of my colleagues to build this consensus effort, what are the things that we can harmonize on and so that you reduce the variability from trial to trial when they may just be subtle differences? And can you have that be a framework? And, in that regard, when you have that foundational framework, it also allows the FDA to say, well, look, there's a group of academic providers who believe that these are the common elements that are really important so that you aren't creating something new for every new study, especially when there's just sort of minor details. I think the same applies to things like eligibility. What does it mean to have relapse refractory disease? How many lines of therapy do you need to receive? Are there things that you can harmonize on, particularly in the early phase setting when the patients that are coming to you have exhausted their ability to receive standard therapies? So I do think along those lines, with a recent study, we struggled with, the time frame from receiving prior therapy. We call that a washout period. Can that be also revised so that there is a commonality? Because from a patient perspective, right, as Tony mentioned, you have to first know what the trial is, know what the eligibility is, and then there's these nuances where you have to be off therapies for 3 weeks or 4 weeks. Can we make all of those standardized? Because I think that would allow us to collaborate more as a community as well.
Those are great points. I love the themes that are coming out in first in human and early phase trials and how we could, obviously safety is the primary focus, but having the ability to collaborate and engage with the FDA early and often, and then building a common framework where we can harmonize certain elements and really learn from prior knowledge is helpful. maybe if I could take us from early development into clinical trial design, and evidentiary expectations for regulatory approvals from pivotal trials. As the cell and gene therapy landscape evolves for therapies like cellular therapies that have had high treatment effects and efficacy potential and unique manufacturing constraints, could we talk about the appropriateness of single-arm trials versus randomized control trials? And from a regulatory and industry perspective, Lola, maybe you could weigh in a little bit here, and then I can ask others too.
Yeah, I'm happy to. I think the first important thing to realize about these cell therapy, these genetically modified cellular therapy products, is that they are very different than the conventional small molecule chemotherapy targeted therapies that most patients with cancer are used to, or at least have heard about. These are therapies that really provide an opportunity to receive a single dose of a therapy that's going to have a lasting, durable effect over a long period of time. And in some patients will cure their disease. And I think that allows us to really think about how do you demonstrate benefit in these therapies in a way that I think is very different than what we're traditionally used to, right? And so I think that the first important consideration when designing a clinical trial, whether you're at the FDA or you're at industry, and even as a patient considers whether to enter a trial, is, do I have a reasonable expectation to benefit from a trial that compares an investigational therapy compared to a treatment that's available that I can get even if I were not on the clinical trial. And so in requiring a randomized trial, the answer to that question has to be yes, right? So you truly don't know which is better. And for whatever reason, you feel you need to have that comparison in order to make a determination that the drug is safe and effective, which is the threshold or the legal requirement to approving a therapy by the FDA. And so for cell therapies, I think we have seen, as Rosanna mentioned earlier, really high response rates in some disease settings, nearing 100% of tumor shrinkage in patients, with a very high proportion of patients achieving a complete response, meaning that there's no tumor seen and those results being durable. And so I think in those settings, one has to consider whether it would be appropriate to randomize a patient to a therapy, an alternative available therapy that may not be providing those types of effects, right? So efficacy isn't the only thing to consider, and sometimes it's very hard to make an assessment of safety in a single-arm clinical trial. But I think that there are opportunities to think about the feasibility of doing a trial that's randomized when you have a high response rate in preliminary efficacy data in a one-and-done therapy, and whether or not patients would be comfortable being randomized to something that is known to provide and deliver inferior results. I think the other thing to consider is really the benefit and how you think about the benefit, because it's not always efficacy and response rate. There are also other benefits that you can get from having a one-and-done therapy where you're not exposed to continual exposure to chronically administered therapies over and over, where you may have complications from that, side effects from that may land you in the hospital, compared to something that where that is a more front-loaded risk, but that over time it may not be the case. So I think that there are opportunities to really think about this case by case, but to also really broaden how we define benefit with the use of these therapies.
Thanks, Lola. I mean, you bring up such great points in terms of ensuring equipoise in a randomized clinical trial when you're, doing that informed consent with patients, and the ethical considerations of randomizing to something that early clinical studies may suggest is not equal, or where you don't have that equipoise. I think for single arm trials, you make an excellent point, is there's always that choice of not to go on those clinical trials and receive currently available therapies. But in the case of randomized control trials, Tony, I'll turn to you, because I think that these are poignant questions around randomization, and from a lived experience, and maybe you can draw from your experience, randomization away from a therapy that has shown early promise can feel unacceptable for patients. Can you maybe provide your perspective?
Yes, from my lived experience, risk benefit assessments for CGTs should include what patients actually go through to receive the therapy. This makes looking beyond responses rates and safety data to things that manufacturing turnaround times, time to treatment, bridging therapy needs, travel burden, caregiver requirements, hospital stays, financial toxicity, and how quickly a patient can realistically access care. For patient, delays are not just dates on a timeline. A delay can mean disease progression, loss of eligibility, more symptoms, more anxiety, and sometimes missing the window to receive treatment at all. Tolerability also matters deeply. Patients need to know how to know not only what side effects may occur, but how long they may last, how they affect daily life, and what kinds of support is available afterwards. In my case, after 12 years of continuous therapy, CAR-T gave me the opportunity to be off treatment. That time off treatment improved quality of life, and freedom from ongoing treatment burden are patient relevant outcomes that should carry real weight and risk benefit decisions.
I love that you talked about patient-reported outcomes and quality of life, Tony, and from your lived experience as well. I think oftentimes, we include some of those endpoints in our clinical trials, but it's variable in terms of how much that supportive evidence helps in the regulatory package that we have for evidentiary standards. So maybe if I can ask the panel, like if we move to this topic of other evidentiary evidence to support a holistic benefit risk assessment, as Tony is alluding to, as cell and gene therapies advance into later phase development, what are the patient relevant evidence endpoints beyond safety and efficacy that could inform the risk benefit assessment for cell and gene therapies? So, Tony, you shared your perspectives. Lola, do you want to share some perspectives from a regulatory and sponsor perspective?
No, I mean, I think Tony is why we do all of this, and I think he was very articulate in really characterizing potential, other benefits that could be really measured to characterize the benefit of these one and done highly effective therapies. I want to be sure to contextualize my remarks and to say that, we also obviously have to be very careful to make definitive conclusions about, efficacy based on very, very, very small data sets, because there's always risk there that patients are very carefully selected and that those results may not translate into broader effectiveness once you start enrolling a broader population that may be more heterogeneous. So just want to contextualize that. But I think that there's real opportunity with cellular therapies to really broaden how we view benefit. I think in other healthcare models where it's a more centralized and comprehensive healthcare model, it is easier to demonstrate really the benefits that Tony was describing around the improved quality of life, the decrease in the loss of time and the ability to go back to work because you're not at the oncologist, every few weeks, either dealing with side effects or dealing with another dosage of your treatment. I think in our healthcare system, it's a little harder to show that those benefits because we do have a very fractured healthcare system with multiple payers who may not see the value in some of this. And that's why I think it's really important that as part of the evidentiary requirement and framework for evaluating the benefits of these treatment, that some of these benefits be more formalized into that process, that it not be sort of subjected, left to the subjectivity of the reviewer, but that it's really incorporated in the legal requirement and the regulatory framework for evaluating these therapies.
Thank you. Thanks for that. I know that we want to leave some time at the end for Q&A, but I wanted to maybe call on each panel member to share some thoughts. Looking ahead, we've talked about a lot of aspects of early phase development, iterative designs, the evidentiary standards that can incorporate comparators versus single-arm trials and patient-related evidence. But maybe if I can ask each of the panelists, what one or two changes in how we design or regulate cell and gene therapies would make the biggest difference in timely, safe patient access to next generation cell and gene therapies. Maybe Nirali, I'll start with you.
Yeah, thank you for that. And thank you for leading this lovely discussion. I think the more we can harmonize and have that foundation to build upon, and the more we have these early discussions with the FDA that can also be not just protocol specific, but that can allow us to have information that is generalizable across cell therapies for specific diseases, that would allow us to be more streamlined and more efficient. I think the other point that I would make is I think it's really important for first in human, first in child cell and gene therapies to also remember who is the patient who is coming to this study. They're coming to the study because all standard therapies have failed, potentially other experimental therapies have failed. And so being mindful that as we think about risk benefit, the alternate may not actually include any therapeutic option. There are patients where no stone has been left unturned, right? And so I think it's really mindful that we think about benefit in that context as well. One of the challenges we've had is that people are so excited about CAR T-cell therapy, the assumption is that they will work equally well in every disease. And so there really is a close relationship between the patients and the providers to make sure that there's full understanding about the specific construct, the experience to date, and where it's going to lead them. But that benefit can come in many ways.
Thanks, Nirali. Just to underscore, I think it's something that that you touched upon, the structured discussion about when and how additional evidence may be expected needs to be taken in the context of where an asset is in its development life cycle. And I think that those, whether a study can proceed in early phase versus the evidentiary standards that are needed for regulatory approval, obviously in later phase are very different. Lola, maybe I can ask you for your one or two wish list items to expedite cell and gene therapy access for patients.
No, I think I would echo what has already been said, and I would just add that there's really the advances in this space are going at a breakneck speed, right? And I think that more understanding of how we can leverage what we have already learned and make these incremental changes to these products, platforms to optimize safety, optimize efficacy, optimize better targeting, to address both of those things, is really important. I think FDA is really moving in that direction. We're really encouraged by their recent guidance around leveraging information. But the information that you can leverage is really dependent on how much we're all willing to share what we know and what we've learned. And so I think my wish list would include a lot of sort of pre-competitive sharing of information and knowledge about our experiences, so we're not doing duplicative work. But I would also say that, I would want to see more opportunities to leverage information, even if it's not, direct data that is coming from other sponsors, to be able to start from a place above where we've already accumulated a lot of information and knowledge, so that we're not wasting time doing things just to check boxes, but that we're really capitalizing on what we've on the learned experience to move faster and move better.
Thank you. Thanks, Lola. Tony, from your perspective, you've gone through, CAR T therapy. What are, one or two changes that you think you were there at the May 6th meeting, you've listened to the panel discussion today. What's on your wish list to expedite the next generation of cell therapies for patients?
Yeah, from my perspective as a patient and advocate, two changes would make the biggest difference. First, create more flexibility and efficient pathways for therapies showing strong early signs of benefit. Patients with serious diseases often cannot afford unnecessary delays. We need rigorous science and safety oversight, but we should also embrace innovative trial designs and risk-based approaches that can help promising therapies reach patients sooner. Second, make patient access part of the conversation from the very beginning. Therapy only matters if patients can actually receive it. Trial design and regulatory decisions should consider real-world barriers such as geographic access, manufacturing capacity, referral pathways, caregiver support, and treatment timelines. As someone who benefited from CAR T therapy after living with multiple myeloma for more than a decade, I know firsthand that timely access can mean for patients, innovation is not just about developing the next breakthrough. It is about ensuring that breakthrough reaches people while they still have the opportunity to benefit from it.
Thank you so much, Tony, for sharing that. We'll move now to the question and answer portion of this webinar, and some selected questions I see in the chat. What are the biggest barriers to ensuring equitable access to these innovative therapies, particularly for underserved and underrepresented communities? Maybe Nirali, do you want to take that one? Because I feel like, especially in pediatrics, I think that oftentimes the pediatric patients are overlooked. And so maybe you can share some perspectives that you have from the academic setting.
Yeah, so pediatric oncology is particularly unique because the vast majority of centers are really academic centers, especially when you're thinking about early phase cell and gene therapy trials. I think Tony pointed this out. I think first you have to have knowledge about what is available. And I think probably there's sort of two elements to it. One is sort of the provider who's providing care for the patient and sort of understanding what is the patient looking for. Are they interested in phase 1 studies? And I think removing any bias that there would be in providing that information broadly to all patients about the availability of all trials, and not putting any emphasis on what they think the patient may want. I think it would be helpful for patients to know the full spectrum of what's out there. Second, I think that if you could do more multi-center cell and gene therapy studies, you'd be able to improve issues related to access. I will tell you, however, that doing multi-center studies for cell and gene therapy, particularly in pediatrics, has been an incredible challenge. These tend to be academic studies. There is generally insufficient funding. Cell and gene therapies are quite unique. You have to think about centralized models of manufacturing, shipping, and doing the delivery. And if we can come up with models that can support that so that children don't have to travel across the country, could at least go somewhere within the same time zone. I think that that would make a huge difference, but that is something that we really continue to struggle with in the pediatric oncology setting.
Yeah. Thanks. Does anyone else want to weigh on this? Biggest barriers to ensuring equitable access for underserved, underrepresented communities?
Yeah, I mean, I think I can add to this. I would agree with everything that Nirali just shared, and also that Tony mentioned before. I would say that in many ways the barriers that we see for non-innovative therapies are just exacerbated in the context of cellular gene therapies, just because of the complexity of the expertise that's needed to really make sure that you're safely managing patients. And so I think we see that new therapies tend to be more accessible in large academic centers, and then they, after some many years, start to permeate into where most of the patients reside. And we see that same effect with these innovative therapies. And I think as we learn more about them, as we develop therapies that are safer and can be administered in an outpatient setting and can be administered, and there's more knowledge about how to manage these toxicities, we will see that greater access. But I think the biggest barriers are the same barriers that we see for other therapies. There are also barriers around just sort of payment for these therapies that are significant issues to address by people who know more about payer models and such. But I think the biggest barrier really is, where does the expertise reside to administer these therapies? And that's often not where most patients are.
Yeah. No, I mean, it's such an important, and it's really the burning platform that we have with innovative therapies like cell and gene therapies that are required to be given at treatments, at specialized treatment centers. And we often grapple with this, and we throw out this statistic a lot, is that despite the advances in the cell and gene therapies that are currently approved by the FDA, still only about 2 in 10 eligible patients are able to receive the cell therapy. And when we look at the reasons for that and break it down, some of it is what you were talking about, Nirali, is that maybe this conscious or unconscious bias about who's eligible for a cell therapy, and that might be patient-related factors, or maybe just unconscious bias about logistics and willingness to travel, or ability to have the supportive care needed to go through a cell therapy, and some of it is just physicians not knowing, and patients not knowing. So I think what I'm hearing here in a call to action for our advocacy partners is raising that awareness and activating the patients so that they are educated on the clinical trials that are available to them. Because I think if no matter where you are, if you go to your physician and you say, hey, what about this trial? Would I be eligible? Then that, will bring the discussion up into the surface instead of, those biases taking control and impeding, efficient access for patients. So, okay, another question. What emerging next generation therapies show the greatest potential to improve outcomes for patients who currently have very limited treatment options, and what milestones are needed to bring these therapies into clinical practice? So maybe Lola, do you want to take that one?
Yes, I can start. I mean, I think there's a lot of innovation in sort of the construct of these cell therapies, as I mentioned before, that are intended to make them safer, to make them more targeted, and to make them more efficacious. I think the other area of innovation is around, how you can develop these products such that they can be a greater proportion of patients can receive them as outpatients. And so that's another area of innovation that I think will improve access. I think the other part of this is really, innovation in manufacturing of these products such that there's not a long lag time from the time for autologous products, which are products that are manufactured using the patient's own cells, that there's a lag time between when those cells are collected from the patient and then taken and engineered and then infused into the patient. That lag time can really be prohibitive for many patients whose disease really is moving and progressing very quickly, and they don't have the time to wait. And intervening treatments, or bridging therapies as they're called, may not be adequate to really address that. And so for those groups of patients, these therapies can be inaccessible. But with improvements in manufacturing that really shorten that time, where you can have a very fast turnaround time of within a couple of days, and then a few more days to make sure that it's meeting the quality requirements and specifications, you can have a broader population that has access to these therapies. I think there's also innovation around rather than doing all this manufacturing outside of the patient, really directly delivering the genetic modification into the patient and having that reconstituted. So having basically the patient be the manufacturer of the cell therapy. And that's really, really promising, because that's going to allow us to really scale these therapies, so manufacture them very quickly and have them be accessible to patients very quickly. But there's a lot, obviously, to learn about the safety profile, and I think there's also a need there to have a greater understanding of what the regulatory requirements and openness to the benefits that these types of innovations will bring to patients.
Thank you. Yeah, indeed, a
Can I-
really exciting field. Go ahead. Oh, go ahead, Tony.
No, I just kind of wanted to add to that, what Lola just mentioned. I think patient communities are being included more than before, but we still need a seat at the table early and earlier and more consistently. Patients can help define what acceptable risk means when you are living with a serious disease and maybe running out of options. And we can also provide important perspective by manufacturing trade-offs, because time is not theoretical for us. Delays can mean additional treatment, disease progression, and even losing the opportunity to receive a therapy. So these realities should help inform these decisions.
That's an excellent point, Tony. I think it's important to have that patient voice in the beginning, especially as these newer innovative therapies are coming in. And when we think about the next generation of cellular therapies, where, as Lola was alluding to, what's called in vivo cell therapies, where the manufacturing goes from, the manufacturing facility to using the patient's body as the manufacturing facility. And it's, it brings up a lot of opportunity in terms of off-the-shelf scalability of delivering cell therapies to patients. But then also with this new technology, there's going to be new sets of questions and regulatory, and how the regulatory pathways that have been used for ex vivo cell therapies, will they still apply to these? I think the answer that we heard at May 6th was yes. And so that's encouraging that we could do that. But I think, stay tuned there. It's obviously a nascent field. It's super early, but there's a lot of excitement. Yeah. Let's see, I'm gonna go through the questions. This is like speed questions now. Oh, Nirali, you'll love this. So does the FDA require experimental therapies for cancer first be tested in adolescents and young adults prior to going to younger children? And what can be done to promote access for children with relapse refractory disease who don't have the time to wait for that data to be generated in adolescents and young adults before them, especially in tumor types where the epidemiology is such where it, does not affect adolescents and young adults.
Yeah, so thank you. It's an excellent question. And to be clear, when we think about pediatric enrollment for early phase therapies, we always have to go back to sort of the regulatory framework and sort of the ethical parameters there. And for studies that involve or that are that have the prospect of direct benefit, there is not a requirement to enroll adults before children, and certainly in diseases where there is no adult equivalent population, you would be enrolling pediatric patients. With that being said, there's sort of been a general approach to say, well, if the disease does exist in a comparable adult population, it would be reasonable for the purposes also of having that adult consent to be able to be provided and then to scale down from children. I think that there's a lot of ongoing discussion about that and trying to see what we can do to enhance or increase pediatric enrollment early on, especially when it is unlikely to find adults who have that disease or that may stall or delay enrollment or development. And I would love to tell the audience to go back to the FDA ARM panel that was held earlier this year that specifically focused on the pediatric age of inclusion, and there was a really robust discussion that is available to look at. I would strongly encourage folks to review that.
Thanks. Thank you, Nirali. Okay, so last question, and I think that, we discussed this, Tony, you talked about, wanting a seat at the table and having a voice earlier on. So what can we do as a community to ensure that the patient voice is incorporated earlier on in clinical trial design, in, manufacturing decisions, et cetera? And so maybe Lola, do you want to take that one first?
About including the patient
Yes, including the patient voice in clinical trial design and earlier on, so that we're ensuring that what we may consider acceptable risk benefit may be different from what they are willing to take or otherwise, and maybe yeah.
Yeah. No, I mean, I think that what it takes is to just do it. I think, both drug manufacturers and the FDA need to start from the patient, right? I think we have to develop products that really are being developed to address patient needs. And that considers not only the needs of having therapy, but also the needs of being able to access those therapies once they are approved. And so you have to start there. And that means, hearing from the patient about how they view access to a trial, to something that's unknown versus what is known, what they would consider a benefit, a meaningful benefit. Sometimes some therapies don't deliver cures, but they may, they may provide respite from a more chronically administered therapy for a certain period of time, and that could be considered a benefit. So defining what that benefit means is important. And I think only the patients can provide that perspective. No one else can provide that perspective. But I think, developers as well as the regulators need to have those conversations and hear directly from the patients about that, because then that sort of feeds into what is a proposal that is acceptable to the FDA. Where do they think they can, implement some flexibility from the sort of a very rigid regulatory framework to account for this patient perspective? But I think it needs to start early, before you design the trial, before you decide what the endpoints are. You have to talk to the patient, and they have to be included all the way, along the way at every stage.
Yeah.
Thank you. Oh, go ahead, Tony. Did you want to add anything?
Yeah, I just want to say I couldn't agree more with that. I just look at the next generation of CAR T therapies, allogeneic, off-the-shelf cell therapies, chimeric antigen receptor, natural killer cells, T cell receptor therapies, all of that gene edited products. And as you were mentioning, the in vivo approaches and expansion into solid tumors, it's just so exciting. What excites me most is not only the science itself, but the potential to make these therapies faster, safer, more doable, and accessible to more patients who need them. Beyond what is available today, I think the next wave would include more off-the-shelf options, smarter engineered cells with safety switches, or multi-targeted designs and therapies that may not require the same long manufacturing wait as a patient. That matters. The future should not only be about more powerful science, but about getting the right therapy to the right patient at the right time.
That is so well stated. I'm going to let you have that last voice, Tony. Thank you so much for so eloquently and so passionately making that case. And to all of our panelists, thank you for your gracious time. I hope that the audience really felt the consistent collaborative spirit that this panel has, and it gives me hope that as regulators, sponsors, clinicians, and patients, we come together, bring that patient-centric voice forward, and ensure that the scientific progress that we create in the lab and move into clinical trials can reach patients safely and efficiently so that they can get the right treatment at the right time. So thank you, everyone, for your participation. And I see that Mark has come on. Maybe you want to say any concluding remarks, Mark?
Just briefly, just thank you, Elena, Rosanna, Nurali, Lola, and Tony, for a very thoughtful discussion. And thank you all for joining us today and contributing questions to the conversation. I am encouraged, much of today's discussion, being focused on solutions, and can say our organization, Friends, we remain committed to continuing these conversations and translating them into practical recommendations that can help move the needle. Thank you again for joining us and for your continued engagement in this work. And I hope everyone has a great afternoon.